Все.
На всякий случай пока ставлю, я Максим предупрежу.
Хорошо. То есть я ухожу и закрываю и сдаю?
Да.
Хорошо, спасибо большое. Good afternoon, ladies and gentlemen. Excuse me for beginning of the lecture, not in time, but topic of today's lecture is immune response or immunity to different types of microbes: bacteria, viruses, fungi, protozoa, and helminths. There are several general features of immunity to microorganisms, mostly general features. Both innate and acquired immunity mechanisms are involved in the immune response, innate and acquired. Microorganisms causing human disease are very different, and to effectively eliminate all of them, specialization of acquired immunity had been developed evolutionarily. Specialization. This specialization is based on at least two major cornerstones. First, different isotypes of antibodies. You remember that there are immunoglobulin G, immunoglobulin M, immunoglobulin A, and immunoglobulin E. And we can add to this list immunoglobulin D. But immunoglobulin D is not a circulating antibody. It means in the circulation of the blood and lymph, immunoglobulin G, M, A, and E. Second basis of the specialization, presence of many different types of T helper cells, of CD4 T helper cells. They are most important among them: T helper type 1, type 2, and T helper type 17. We can add to this list two more, at least two more. This is a T follicular helper, FH, and the T regulatory helper cell. But the last one, regulatory, mostly causes down-regulation of immune response, immunosuppression. If latent or persistent infection is formed, you know this is infection when microbe occurs in the human body during a long time, immunity may control microbes, replication of them, but cannot eliminate them. And in these conditions, when persistent infection is happening, balance between immune mechanisms and bacterial capacity to survive is maintained in the human body. And this balance is very important to keep bacteria and viruses in non-harmful state. If many diseases, tissues, injury, if many diseases, injury of tissues is caused not by microbes, but by the immune mechanisms. What at least in the T helper 1 immune response against intercellular parasites of different origin, intercellular bacteria, intercellular viruses, what this cell damaging may be caused by T killer cells and macrophages, stimulated by T helper 1 to destroy cells infected by microbes in this way, to destroy factories for production of new parasites. Now, let's start from the immunity to bacteria, namely to extracellular bacteria. Now, extracellular bacteria, they are organisms that occur usually outside the host cells. For example, Staphylococcus, Streptococcus, Escherichia coli. Various species of extracellular bacteria may cause diseases via two principal mechanisms. First mechanism of the generation of the disease by extracellular microbes is generation of inflammatory reaction. You remember that inflammation, this is a reversible changing of the circulation of the blood in the infected place. If disease is generalized, this inflammation with the increased vascular permeability is happening in all the human tissues and on all the human vascular system. Second exception for inflammation This is toxin productions. Toxins are poisons caused by, no, generated by bacteria, produced by bacteria to damage human cells or to affect the interaction of human cells. You remember that toxins are subdivided to endotoxins and exotoxins. Endotoxins are lipopolysaccharides of the gram-negative bacteria. Exotoxins are very different. The variety of microbes that may, variety of poisons, exotoxins, they're proteins mostly, that may cause cytotoxic effects to kill human cells, or they have mechanisms to interfere with normal cellular function without destroying of cells, but causing abnormal function. And some toxins act via stimulation and production of the cytokines. The cytokines are the regulatory molecules produced by human cells that usually regulate activity of other human cells. But improper producing of cytokines causes very severe problems, because cytokines, if produced with very high concentration, interleukin-1, for example, and interleukin-6, may cause inflammation that may damage tissues and cause even lethal results. Innate immunity to extracellular bacteria is associated to many factors. Very important mechanisms are complement activation, what in the lower left part of the slide, complement activation, you see lectin pathway initiated from the left upper group of factors. Classical pathway, when first complement molecule activated, is C1, and alternative pathway, when first activated molecule, is C3. Finally, all these three pathways, they finally cause the cleavage of C3 molecule, and after that, via producing of C5b and attaching to C5b on the membrane of the bacterium, C3, 7, 8, and 9 components cause the membrane attack complex, causing lysis of bacteria. This is a typical humoral mechanism. In the context, phagocytosis, what here you see in the right part of the slide, dendritic cell. Dendritic, why this cell I think is dendritic? Because it has filamentous processions of the surface. They are used to contact to many cells simultaneously. Phagocytotic effect, this is engulfment of microbe, digestion of this microbe, and eliminating of microbe in this way. Immunity to extracellular bacteria associated to activation of phagocytosis and inflammation. Phagocytes express many surface receptors for foreign molecules. They are named pattern recognizing receptors, you know. The ligands for these pattern recognizing receptors of human cells are pathogenesis-related molecular patterns, or PAMPs. Receptors for these pattern recognizing receptors, they are variable. No, some of them are shown here. You learned that before. This is a mannose receptor, or CD206. Scavenger receptors, or CD36, that recognize bind to the lipopolysaccharide and oxidize lipoproteins, lipotecoic acids. FC receptor recognized extracellular bacteria opsonized by immunoglobulin M or immunoglobulin G. FC receptor, this is receptor that binds to the FC portion of immunoglobulin. And this immunoglobulin, if binds to the antigens of the surface of bacteria, what makes visible for phagocyte with this presence of the pumps that react to the FC receptors. Then complement receptors receptors for C3b molecule of the complement system. C3b molecule binds to the many bacteria, and complement receptors recognize C3b molecules that bind to the bacteria and recognize these bacteria, and after that phagocytose them. Very important receptors of the pattern recognizing receptor system are Toll-like receptors. There are many, at least 11 different Toll-like receptors are known, have been described in human and animal cells. They may recognize variety of absolutely different substances. Lipopolysaccharides, take away cases, single-stranding RNA, single-stranded DNA, or double-stranded RNA, and other types of the unusual nucleotide sequences. What 6 receptors shown here is Minkel receptor. This is an abbreviation consisting of following words: macrophage inducible calcium dependent C-type lectin. This is a protein recognizing carbohydrates on the surface of the some of the bacteria. Minkel receptors recognize glucose and Tihalose mycolates and the Carinobacterium or Mycobacterium. It means the causative agents of tuberculosis and causative agent of the diphtheria. Minkel macrophage receptors. Some human cells surface receptors, PRR, pattern recognizing receptors, are capable of recognizing fungal, bacterial, helminth, and virus pathogen-associated patterns. But here you see that fungal pumps may be recognized by dictin-1 receptor. This is a transmembrane protein that, after recognizing of the fungal and bacterial pathogen-associated molecular patterns, transmit the signal to the cytosol of the cell and initiate realization of the signaling pathway that makes cell recognizing this microbial pathogen of producing of the proteins, for example, cytokines. It means dictin-1 receptor may recognize some fungal pumps and bacterial pumps. Dictin-2 receptor, SNX, also of the dictin family, recognizes fungal products, usually polysaccharides, bacterial polysaccharides, and polysaccharide soluble components of helminths. Minkel receptors, you know that they are macrophage-associated lectin receptors. They recognize fungal and bacterial pumps among bacteria, first of all, mycobacterial pumps. And the last receptor, the blue color, this is a C5A blue receptor. This receptor is named C-type lectin domain family 5 member A. Very complex name, but what is interesting? This receptor recognizes such viruses as the influenza virus or dengue virus. What this? CLE-C5A. And after recognition, cells generate signal that activate transcription and translation of the pro-inflammatory cytokines, tumor necrosis factor alpha, interleukin-1, interleukin-6, 8, interleukin-17A. These cytokines cause generalized inflammatory process that, in case of the dengue fever, is named the syndrome that is named dengue virus-induced hemorrhagic fever. A hemorrhagic fever, this is a typical classical manifestation of this severe virus infection. It means, in this case, what this virus C-type lectin domain family 5 member A receptor initiates pathway that finally will make cell of producing pro-inflammatory cytokines, causing dramatic changes in the vascular circulation of blood. In this scheme, you see many different variants of the activation of the innate immunity in the cell that is shown in the left part of the slide. Now, first of all, some extracellular bacteria that shown in the right part at the top, they may activate mannose binding protein that may, after that, stimulate complement activation by the lectin pathway. Some microbes or microbial products may activate C1 component of complement system, activating classical pathway, and synthesis of C-reactive protein. This is acute phase protein that is increasing the concentration very intensively in case of the bacterial infection. Result of activation of these three proteins is generated of inflammation. Viruses may infect cell, injecting virus nucleic acid to the cytosol. This double-stranded RNA or single-stranded virus RNA that in the cytosol are recognized by pattern recognizing receptor of the RIG type. RIG, RIG1 type receptors recognize this molecule, initiating development of signaling pathway that after stimulates NFKB and FKB. This is nuclear factor kappa B, nuclear factor that controls process of transcription of some genes. For example, genes that code for interleukin 1 and interleukin 8. They are pro-inflammatory cytokines that also cause inflammation. Mannose receptor, what this brown in the center, or Toll-like receptor, some of them Toll-like receptor 4, Toll-like receptor 1 and 2, they recognize lipopolysaccharides and flagellar proteins of the some bacteria, also generating signaling pathway causing activation of nuclear factor kappa B and transcription and translation of the interleukin 1 and other pro-inflammatory cytokines proteins. Toll-like receptor 3, 7, and 9, they are exposed to the vesicles, endocytotic and phagocytotic vesicles. They react to the single-stranded RNA, double-stranded RNA, and also activate the certain signaling pathways, finally causing production of the pro-inflammatory cytokines and generation of inflammation. Well, this inflammation generated by complement activation and the interleukin 1, 8, and some other interleukin activation causes swelling of tissues, increase in the vascular permeability in tissues. Some of these substances recruit the leukocytes, for example, neutrophils, and they act as the chemoattractants for these cells. They enter to the area of the inflammation, causing leukocyte infiltration. And these leukocytes, phagocytose microbes and cause damaging of the infected cells, for example, cells that are shown here. Innate immunity to extracellular bacteria. Innate immunity to extracellular bacteria is associated to phagocytosis. What phagocytosis of the bacteria shown here, phagocytosis in the neutrophils that are attracted from the vessels to the affected area. Innate immunity is associated to activation of complement system, at least by the alternative pathway and lectin pathway. These immune reactions associated to histamine release from the mast cells histamine, changing vascular permeability, increasing the transport of water, blood plasma, and cells of the blood from the vessels to the tissues, causing local inflammatory reaction and swelling of tissues. In the process of the innate immunity, usually pro-inflammatory cytokines are produced by macrophages, dendritic cells, and some other cells. The most important pro-inflammatory cytokines, are tumor necrosis factor, interleukin-1, interleukin-6, and interleukin-23. This is not a complete list of the pro-inflammatory cytokines. This list may be made longer. For example, we can add to this some interleukin-13, interleukin-12, and some other also. Adaptive immunity to extracellular bacteria. This slide differs from previous because demonstrates adaptive. It means acquired immunity to extracellular bacteria. The major mechanisms of the adaptive immunity is associated to antibodies. Antibodies are produced by plasma cells. Plasma cells are matured forms of the B lymphocytes. B lymphocytes recognize microbial antigens by the membrane-associated immunoglobulin M or immunoglobulin D. After that, B cell activation also depends on the cytokines of the T helper cells, in this case of the T helper type 2. When antibodies are produced, they bind bacteria causing their neutralization. The antibodies may cause inflammation, for example, activate complement system. They involved in the opsonization, this is facilitating phagocytosis. And in the lower part of the slide, you see on the left toxins, or these triangles, contact to the antibodies. Antibodies neutralize these toxins, and toxins cannot affect the cell. The next, C1 complex. With yellow C1 protein binds to the Fc fragments of antibodies that were before absorbed to the antigens at the surface of bacteria. Complement is activated here, and after that, this bacterial cell may be damaged. The next, effector cell, what you see it on pink cloud, and this is a symbol demonstrating such cells as the natural killers. Natural killer also has Fc receptor for immunoglobulins G and M. They these effector cells, natural killers, via antibodies recognize surface of the target cell, attack this cell by the release of the cytotoxins. No cytotoxin produced by natural killer cell are perforins and granzymes. And the last small object shown here, this is a phagocyte that engulfs the opsonized bacterium. Blue bacterium binds to the antibody, antibody binds to the Fc receptor phagocyte, and phagocyte engulfs this bacterium more intensively. With this more intensive engulfment is the realization of effect of opsonization. When say about cellular components of the elimination of intercellular bacteria, we have to say about CD4 cells, first of all. CD4 cells, they are critically important cells to activate immune response to extracellular bacteria. What here you see on the left part of the slide, dendritic cell that presents bacterial antigen to the naive T helper cell, recognizing this microbial antigen by the T cell receptor. T helper cells involved in the struggling against extracellular bacteria are mostly T helpers type 17 and T helpers type 1. What about, we say now? We say now about cellular immune response against extracellular bacteria. Before we talked about humoral immune response shaded to production of antibodies, and now we see about cellular immune response. T helper cell activated by the antigen presented by the antigen presenting cell may be matured by two pathways. First is transform into T helper 17 effector cell. T helper 17 produces such cytokines as the interleukin 17. By the way, T helper 17 is named because is the major producer of the interleukin 17. It means T helper cell is named after interleukin synthesized by this cell. Exception for interleukin 17, T helper type 17 cell may produce interleukin 22, tumor necrosis factors, other cytokines. But these cytokines were not produced in the rather high concentration. They activate macrophage and activate neutrophils. They activate influx of macrophages and neutrophils from the vessels to the tissues, infected tissues, where macrophages and neutrophils participate in the development of the inflammatory reaction. For example, macrophages engulf opsonized microbes. Neutrophils also may engulf opsonized microbes, and macrophages and neutrophils release reactive oxygen species that destroy microbes and microbe-infected cells. The next T helper cell is T helper type 1. Second line of the T helper cells of the maturation is maturation of T helper 1 cells. This T helper type 1 cell produces in interferon gamma, interleukin 2, and tumor necrosis for alpha and beta. Interferon gamma synthesized by T helper type 1 superactivate macrophages. Such the macrophages activated by T helper 1 cells by interferon gamma. They phagocytose microbes more intensively. They digest microbes more intensively because activated macrophages start producing of the many reactive oxygen species, including nitric oxide. Nitric oxide is produced only by macrophages activated before by the interferon gamma. You see two types of events. T helper 17, they attract macrophages and neutrophils to the infected area. T helper type 1 superactivate macrophages to activate the digestive and engulfment capacities. And substances released by macrophages are involved in the inflammation additionally. Activated macrophages produce interleukin 1, interleukin 6, and interleukin 12, and interleukin 13 that participate in the development of the inflammatory reaction. It means extracellular bacteria are controlled by antibodies, T helper type 1, T helper type 17, by the complement activation, and many other mechanisms. Here you also see adaptive immunity to extracellular bacteria. No, extracellular, it means bacteria that occur outside of the host cell. Not inside, but outside, in the space between host cells. What even do you see here? This slide is divided into halves, upper and lower. In upper part, you see that bacteria activate B cells that have antibody receptors specific for antigens of the bacteria. When this B cell is additionally activated by T helper cell, this T helper cell is T helper type 2 cell. With interleukin 4 and 5, they are not shown here, but you have to understand that they're T helpers type 2. They additionally activate B cell, previously activated by the antigen, and this double activated B cell start replication, and finally most of these descendants of the B lymphocytes are transformed to the plasma cell. Plasma cell stops the replication, produces antibodies, and these antibodies bind to the bacteria, causing the agglutination, bind to the special Fc receptors of macrophages that engulf this bacteria more intensively. This is the effect of sonization. Neutrophils involved in the inflammation also may be activated by products synthesized in the result of the complement activation, because complement activation causes synthesis of C5a and C3a molecules that are hematocrit transfer macrophages and neutrophils. And finally, complement activation causes lysis of the bacteria. In this upper part of the slide, you don't see T cells. You see only what's helper type 2. In the lower part of this slide, marked by the letter B, you see how bacteria are engulfed by the antigen-presenting cell, for example, dendritic, most active among these cells. This cell presents microbial antigen to the T cell receptor of the CD4 helper T cell, and this helper cell may be matured to different effector cells. For example, this helper cell, when is the producer of interleukin 17, is matured to the T helper 17 cell. This cell causes neutrophil inflammation, attracting neutrophils from the vessels to the infected area. If T helper cell would be matured to the immunocompetent cell producing from gamma, we will recognize this cell as the T helper type 1 that hyperactivates macrophages. And if you see that T helper cell finally causes activation of the antibody synthesis by plasma cell, you will understand that mostly this T helper cell is T helper type 2. But even this cell, we can put what in this upper position where T helper cell cytokines are shown as the stimulators of the plasma cell synthesis. Adaptive immunity, extracellular bacteria, development of this adaptive immunity may cause negative reactions. Inflammatory process activated by immune response to extracellular bacteria may cause the damaging of the human cells, and if inflammation is systemic and pro-inflammatory cytokines produced by the macrophages, dendritic cells, interleukin 1, 6, interleukin 13, if they are prevalent in the bloodstream, septic shock may be developed. Septic shock, this is a syndrome where changing of the vascular permeability increasing finally causes fall of the blood pressure. This fall of the blood pressure may, of course, with itself cause the inadequately low transport of oxygen to the many organs, to kidneys, lungs, brain, causing unconsciousness and very severe sequelae. Inflammation caused by realization of adaptive immunity processes may cause damaging of tissues and secretion of pro-inflammatory cytokines, what's shown here, tumor necrosis factor, both alpha and beta, interleukin 1, 6, interleukin 23. They stimulate not only vascular permeability increasing, but producing of active phase proteins, for example, C-reactive protein in the liver. These things also can cause such clinical manifestation as the fever, headache, chills, rash, what you see, skin rashes in the patient. It means inflammatory reactions by stem cells after activation of the adaptive immunity may cause the complications, negative results. In majority of cases, the self-limiting processes when patients heal, but in some cases, very deep and intensive damaging of tissues may cause even lethal complications. The next type of bacteria that we have to discuss are intercellular bacteria. What are the distinctive features of the intercellular bacteria? That can survive within phagocytes. It means macrophage may engulf this bacterium. After that, bacterium is not ruptured and replicate inside of the phagocyte. Second, intercellular bacteria are inaccessible to circulatory antibodies because they are protected from antibodies by the cytoplasmic membrane of the host cell. Elimination of the intercellular bacteria is the function of the cell-mediated immune response mostly. It means that most important mechanism of the struggling against intercellular bacteria associated to the cell-mediated immunity. That is not the most important to eliminate the extracellular bacteria. But in this case, this would be most important mechanism to eliminate intercellular bacteria, cell-mediated immunity, with the involvement of the T helper type 1 and cytotoxic CD8 positive T lymphocytes. In many infections caused by intercellular bacteria, the host immune response causes tissue injury. In cell-mediated immune response, tissue injury may be caused by hyperactivated macrophages or by the cytotoxic T cells. Innate immunity mechanisms in the struggling against intercellular bacteria are phagocytosis by the neutrophils and phagocytosis and activation of the macrophages, not macrophages. Third cell involved in the innate immunity to intercellular bacteria are natural killers. Natural killer cells kill the host cells infected by the cellular bacteria and viruses, of course, and other intercellular parasites, fungi. And the natural killers, they demonstrate their activity in the first few days after infection. It means before appearance of antigen-specialized cytotoxic T lymphocytes. It means natural killers, even compared to other killer cells, CD8 cytotoxic lymphocytes, they act quicker. And natural killer cells, after activation, may secrete interferon gamma. And this interferon gamma may activate macrophages for digestion and engulfment of the cells infected by the bacteria or viruses. And they, via synthesis of interferon gamma, I mean natural killers, in this way enhance a process of the maturation of naive T helper cells, CD4 helper cells, to the T helpers type 1. But these T helper type 1 cells, they are not shown here because they are cells of the acquired immunity. You know that natural killer cells, they may recognize activating signals. What in this figure you see in the left part of the space between natural killer and low situated infected cell, target cell. You see three activating receptors of the brown color that activate three multiple activating receptors. It means ligands for activating receptors brown activate receptors, and these receptors transport signal to the cytoplasmic enzymes. Phosphorylation of protein tyrosine kinases happen. What here you see phosphorylation of three protein tyrosine kinases. These kinases are phosphorylases. They phosphorylate the next adaptive protein that transports signal to the nucleus, and in this way initiate the synthesis of the proteins involved in the killing of the target cell. In the contrast, from other side, the surface of the natural killer cell occur inhibitory receptors. Here you see only one inhibitory receptor of the green color. Inhibitory receptor recognizes MHC protein type 1. This is a major histocompatibility complex protein type 1 that is itself independently on the microbial antigens that may be presented by this molecular node. MHC1 protein is itself activates inhibitory receptor and generates signals that counteract to the activation signal for natural killer. But in our skin, protein tyrosine phosphatase with this green protein neutralizes one of the phosphorylation acts, but you see that two more phosphorylation processes are not blocked, and what really this natural killer cell has three activating signals and only one is neutralized. It means two pass to the nuclear ligands and cause activation. Activated natural killer cell that is activated by the missing of self, it means not enough expression of MHC protein type 1 on the surface. Natural killer accumulate vesicles, so-called granules, contain perforins and granzymes below the cytoplasmic of natural killer. After that, these granules are degranulated. It means membranes of these granules fuse to the membrane cytoplasmic of natural killer, and content of granules attack, becomes exposed and attack surface of the target infected cell. Perforins are integrated to the membrane, cell membrane of the target cell, and cause the pores. Through the spaces of these pores, holes of these pores, perforins, the hydrolytic enzymes, they pass inside of the cytoplasm and nucleus, causing damaging of these cells. Additionally, natural killer, when it's activated, that is shown here, started producing interferon gamma. Interferon gamma activates macrophages for more active phagocytotic damaging of the phagocytosed bacteria. Substances that additionally activate natural killers are interferons of the first type, alpha interferon and beta interferon, and interleukin 12 and interleukin 15. With these cytokines actively enhance attack of the natural killers against infected cells and produced by these killers of the interferon gamma. Adaptive immunity to intercellular bacteria is mediated by T cells. Most important of these T cells are two types of cells: CD4 positive T cells and CD8 positive cytotoxic T lymphocytes. In this slide, you see the top part of the slide, upper, on the left angle, you see antigen presenting cell. Really, this is a dendritic cell that contains bacterium in the phagosome and bacteria in the cytosol, or cytoplasm. It means this is not only antigen presenting cell, this is infected cell additionally. This cell presents microbial antigens to CD4 helper cells and CD8 cells. MHC2 protein presents microbial antigen to T cell receptor of naive CD4 helper cell, or TH0, naive cell. If this CD4 cell additionally gets activated stimulus through the co-receptors, they're shown here. It means co-receptors are CD40 receptor of the antigen presenting cell binds to the CD40 ligand of the helper cell, and B7 receptor of the antigen presenting cell binds to the CD28 receptor of the T helper cell. You see antigen via antigen stimulation of T helper via antigen presentation and via establishing of the bridges between co-receptors of the dendritic cell and helper. This helper cell acquires the third signal also. This is a activation of the T helper cell by the interleukin 12. Here you see arrow with the interleukin 12. This is a cytokine produced by activated antigen presenting cell by the dendritic cell. Triple signal is acquired by this T helper from the antigen recognition, from the co-receptor interaction, and from the soluble signal, interleukin 12. CD4 naive cell, if is situated in the lymph organ where interferon gamma is present in the rather high concentration, start to be transformed to the T helper type 1. This T helper type 1 is not shown, but you understand that this is descendant of this helper T cell that is stimulated by interleukin 12, co-receptors, antigen recognition, and by the interferon gamma that creates the environment causing transformation of descendants of T helper cell to the T helper type 1. T helper type 1 effector helper that controls the acquired cellular immune response. How this acquired cellular immune response is realized? On the top you see purple naive CD8 T cell. This is a precursor of the cytotoxic lymphocyte. If this precursor, CD8, recognizes via T cell receptor microbial antigen specific to this T cell receptor presented by MHC1 protein, major histocompatibility complex protein type 1, not type 2, but type 1. This signal is the first polarizing signal for naive T cell. After that, second polarizing signal, binding of the co-receptors of the antigen presenting cell and co-receptor ligand of the naive T helper cell, in a naive CD8 killer cell. And if this naive cell acquires this third signal in the form of the interferon gamma that is not shown here, and interleukin 2, this is a cytokine secreted by the T helper cell type 1, but in this case naive CD8 cell replicates intensively, and the descendants of this replication are matured to the differentiated cytotoxic T lymphocytes. These matured cytotoxic T lymphocytes start producing interferon gamma. You remember that initially the natural killer cell also produce interferon gamma. And this interferon gamma causes the background with the prevalence of interferon gamma. In this situation, new CD4 helper naive cells also becomes transformed, matured to the T helpers type 1. It means adaptive immunity to intercellular bacteria is mediated by CD4 T helpers type 1 and CD8 cytotoxic T lymphocytes that intensively kill bacteria and virus infected cells. Now here you see the similar situation. I wouldn't comment this additionally. What's shown in this slide? In this slide you see CD4 T helper cell cooperation with CD8 T cells in the defense against intracellular bacteria. Now, for example, Mycobacterium tuberculosis or Listeria monocytogenes. What do you see here on the top left angle? You see macrophage infected by bacteria. What one bacterium is present in the phagal lysosome, but two bacteria, they bypass digestion in the phagosome and could pass to the cytosol. First reaction, MHC1 protein, what this? Stated in the low part of this macrophage, activates T cell receptor via the antigen presented by MHC class 1. Core receptor interaction is happened also. CD4T of macrophage acts to CD4T ligand of T helper cell as B7 receptor of macrophage binds to the CD28 receptor of the T helper cell. T helper cell, naive, replicates and descendants is transformed to the T helper type 1. Why? Because these T helper type 1 cells may be produced in the presence of the cytokine stimulating these cells, and first of all, the presence of interferon gamma. Finally, T helper type 1 cells, they also release interferon gamma. Interferon gamma stimulates what this infected macrophage to destroy phagocytosed bacteria, and these phagocytosed bacteria are ruptured, lysed. But interferon gamma cannot cause the damaging of the intracellular bacteria that is situated in the cytoplasm of the macrophage. But to eliminate what these bacteria, at the same time, naive CD8 cell recognizes antigen presented by the macrophage, infected macrophage, by the MHC1 protein, and in the presence of the positive co-stimulation, these naive CD8 cells, and after activation by the interferon gamma also, and by activation of interleukin 2, both these products are produced by T helper type 1, all these activated naive CD8 cells replicate, and their descendants are transformed to the completely differentiated CD8 cytotoxic T lymphocytes. These T lymphocytes, via antigen recognizing receptor, recognize antigen presented by MHC protein type 1 and attack infected cell. This attack was by the perforin enzymes or conducting of the apoptosis. What this bacterium-infected macrophage becomes disrupted, broken, disappears, and bacteria that occur in the cytosol of this cell also becomes destroyed. At least you see here a process of cooperation of CD4 helper cells type 1 and CD8 cytotoxic T lymphocytes the process of the eliminating of the extracellular that occur in the phagosome and intercellular bacteria that occurred before the cytosol. To kill them, this intercellular bacteria, we have only one chance. This is a chance to destroy infected cells that serve as the carrier and protector of this bacteria. Viruses. You know that viruses are obligate intracellular parasites. Viruses may occur as the virus particles, so-called virions. They are ectocellular viruses. They may be eliminated by antibodies. And intracellular viruses that occur in the cytoplasm of the affected host cell. Of course, antibodies cannot eliminate with this intracellular type of viruses. Now you know that viruses have the unique method of replication, disjunctive method. When one virus infects host cell, after one replicative cycle that lasts 2-3 days, or in some cases 1 day, hundreds, in some cases thousands of progeny viruses are released. What you see here, this progeny orange virus is released from this dark brown cell. Now there are two most important features of the viruses. Let's see how is organized antivirus immune response. Immunity viruses may be subdivided into the innate and the acquired. Now you remember, innate and acquired, this is a typical classification of all immune responses against all the known parasites. Innate immune response includes type 1 interferons. Here you see on the top interferon alpha. This is one of two type 1 interferons. Second point, natural killer cells. Here, natural killer cell is shown that the cell produces interferon gamma in our scheme. Third position. This is a position of the acquired immunity mechanism, neutralizing antibodies. Here in this figure, neutralizing antibodies are not shown, and you understand that they may mostly bind to the extracellular virus or to the surface of virus-infected cell. And the last option here is cytotoxic T lymphocytes. Cytotoxic T lymphocyte also is not shown here. But what do you see in the scheme exception for all these four options? You see CD4 cell, or this gray. This CD4 cell, when it's involved in the adaptive immune response, mostly is the T helper type 1 cell. What then you see that what naive help is transformed to the T helper 1 cell. To be transformed to the T helper 1 cell, descendant of this naive cell has to be activated by antigen via MHC2 protein. Maybe signal is confirmed by reaction of co-receptors. Naive T cell must be activated by the humoral signal by interleukin 12, secreted by antigen-presenting cells, for example, dendritic cells, and by macrophages. And additionally, T helper cell type 1 needs to be activated by interferon gamma that may be secreted by other T helper type 1 cells, matured, immunocompetent, or by the activated natural killer cell that is shown here, the producer of two arrows demonstrating interferon gamma. Principal mechanisms of the innate immunity against viruses by principal mechanisms of the innate immunity, pay attention. Now they are, of course, natural killer cells and synthesis of interferon alpha and interferon beta. Now, how does that natural killer cell, you know? natural killer cell, kills the virus-infected cell infected by the viruses if number of the activating ligands is more than number of the ligands for inhibitory receptors. Interferon alpha and beta, they're produced, synthesized by the virus-infected cell. They are secreted by a virus-infected cell. They interact with the receptors for interferons alpha and beta of the neighboring cells. Bind of interferon alpha and beta to the interferon receptor initiates realization of the signaling pathway that activates the certain portions of DNA that code for so-called antivirus proteins. That proteins are synthesized in the cell, accumulated in the cytoplasm, and when new virus tries to infect this protected by interferon cell, with these antivirus proteins bind to the components of the virus and block them. What most important part of this antivirus proteins, they are blockators of the RNA DNA polymerases. In this scheme, you may see how it's realized in principle. Interferon activating pathway. You see virus with yellow that may be engulfed with the phagosome or may pass through the membrane directly if fusion is happened. Virus also may appear in the cell if cell phagocytosis, the virus-infected cell, and after lysis of the phagocytosed cell, virus nucleic acid may be injected to the cytoplasm. In any case, virus products, for example, virus RNAs and DNAs, may be recognized by toll-like receptors. Toll-like receptors type 7, 8, 9, and 3. They recognize nuclear pumps, pathogenic molecular patterns, mostly they are double or single-stranded RNA molecules or DNA molecules of viruses. In the cytosol, nuclear acids of the viruses may be detected by the Rig-like receptors and node-like receptors. But here you see three variants of Rig-like receptors: MDA5, Rig1, and DAI. These receptors bind to the virus nucleic acids, and altogether with all these Toll-like receptors and Rig-like receptors, they activate transcription factors, for example, nuclear factor kappa B, that activate transcription from the genes that code for interferons, and the cells start to produce interferons, interferon alpha, interferon beta. It means here you see principal moments, mechanisms of the realization of the interferon inducing pathways. Now, how natural killer cell kills virus-infected cell, you may imagine one comment. Some viruses infecting host cells cause inhibition of synthesis of the MHC proteins class 1. And on the cell disappear MHC1 proteins and disappear capacity to activate inhibitory receptors. And inhibitory receptors are not involved in the blockade of the activating signals to the natural killers. The natural killers attack this cell. It means natural killer cells may recognize and kill all virus and bacterial infected cells that don't display in the surface MHC1 proteins. Some viruses, they prevent synthesis and displaying of MHC1 proteins to avoid attack of the cytotoxic T lymphocytes that require representation of the antigen in the complex to MHC1. But at the same time, with these viruses, for example, HIV viruses, preventing synthesis of MHC1 becomes targets for natural killer cells. It means all the diapason of the intercellular parasites may be subdivided into parts. One, they are parasites that don't prevent synthesis of MHC1 proteins, so these parasites are eliminated, and parasite-infected cells, by cytotoxic T lymphocytes. Second group, they are parasites that cause blockade of MHC1 synthesis. They bypass cytotoxic T lymphocyte activity, but they become clients and targets for natural killer cells. Well, this situation is typical for patients with a HIV infection, human immunodeficiency virus disease. Adaptive immunity to viruses is mediated not only by cells, but by neutralizing antibodies also. It means adaptive immunity, not innate. Adaptive immunity for viruses is based on the synthesis of the neutralizing antibodies and production of CD8 cytotoxic T lymphocytes. How to act neutralizing antibodies? They may bind to the viruses in the extracellular space and block capacity of viruses to bind to the surface of the target cell. And this way, target cells becomes protected by these antibodies that bind to the virus. Here also you see how 4 antibodies bind to the antigenic receptors of the virus. And here you see in the photograph part of the slide in the center how antibody binds to the virus that tries to penetrate cell. In the lower part of this photograph, you see how antibody binds to the virus in the phase of the budding. Immediately before budding will be followed by the appearance of the progeny virus, and this virus from the beginning of its life becomes blocked by the antibody. But antibodies cannot interact with the internal components of the virus. What killing of these infected cells, this is a matter of the differentiating cytotoxic lymphocytes. They are descendants of naive CD8 cells. You just know how the cells do act. Antibodies may interact to us inside of the human body and outside. For example, immunoglobulin A may neutralize viruses that occur in the lumen of the hollow organs of the respiratory tract or digestive tract, preventing absorption of viruses to the host cells. Additionally, antibodies, for example, immunoglobulin G and M, may exonize viruses, and these exonized viruses, coated by the antibodies, becomes engulfed by the phagocytes. And viruses that carry antibodies that are bounded to the viruses may initiate activation of complement system by the classical pathway, and complement activation would be happening on the surface of virus, and virus would be lysed. But in this figure you see how viruses may be eliminated by by CD8 lymphocytes and CD4 lymphocytes. In principle, this scheme is very similar to the scheme of the elimination of the intercellular bacteria. Intercellular viruses, the antigens, are presented to the CD4 naive T cells, and if co-receptors support this signal, in the presence of interferon gamma, these CD4 naive cells mature to the CD4 helper type 1. CD4 helper type 1 produces interferon. This interferon stimulates macrophages to destroy phagocytosed macrophages, phagocytosed viruses. Cytoplasmic viruses and their components are not accessible for this signal, but they are targets for naive and after that matured CD4 cells. Naive cells recognize MHC1 protein presented antigen after co-reception and after stimulation by interferon gamma and interleukin-2 secreted by, again, T helper type 1. Naive CD8 cell is transformed to the differentiated cytotoxic killer cell, and this cell recognizes virus antigen in the complex to MHC1 on the surface of the virus-infected cell, and perforin enzyme attack destroys this cell and at the same time destroys all the viruses inside. Now here you see that some cases dendritic cells, phagocytoses, and macrophages also, virus-infected cells, dendritic cells only, not macrophages, but dendritic cells may digest virus-infected cells and may, after that, present the virus antigen in the complex with the MHC1 protein. It means this presentation to the CD8 cells may be done by the virus-infected macrophage. But dendritic cell may present antigen with the MHC1 protein even if dendritic cell is not infected by the virus. It means healthy dendritic cell may cause this presentation. This presentation of antigen to the CD8 cell by the healthy antigen presenting cell, healthy dendritic cell, is named cross-priming. It means cross-priming. This is a presentation with the MHC1 protein complex to the CD8 cells if antigen presenting cell is not infected by itself. Now you understand that naive CD8 cells replicate, their descendants are transformed to the killer cells, T killer cells, and they destroy virus-infected cells in the body. This slide we will ignore. This also. And now about some mechanisms of immune evasion by viruses. Immune evasion. Some viruses may bypass immune response by the antigenic variation, what influenza, rhinoviruses, HIV virus. It means they changed antigenic determinants, and these determinants can be recognized by antivirus antibodies and T cell receptors of the T helper and T killer cells. Second mechanism of bypass, inhibition of antigen processing. Some viruses, they may block so-called TRAP transport system that transports endogenous antigens from the cytosol to the rough endoplasmic reticulum. What blockade of this pore system of parins, so-called TAP, prevents the formation of the MHC1 antigen virus peptide complex. This is this peculiarity has been described for some herpes viruses and cytomegalovirus. Third, production of cytokine receptor homologs. Some viruses code for proteins that serve as the homologs of the receptor for cytokines. It means vaccinia virus and pox viruses producing analogs of the receptors for cytokines may bind interleukin-1 and interferon gamma, removing these cytokines from the infected area of the organism and preventing development of the formation of the cytotoxic T cells. Fourth option, production of immunosuppressive cytokines. Some viruses code for example, Epstein-Barr virus is also herpes virus, code for interleukin-10-like protein. They infect human cells, and the cells start producing interleukin-10-like cytokine. Interleukin-10 is the immunosuppressive substance. It means infected cell produces interleukin-10 analog, and in this way downregulate immune response. It means prevent synthesis of the new helper cells and new killer cells. And finally, some viruses, for example, HIV, human immune deficiency virus, causes death of the virus-infected cell by itself. And as a result, CD4 T cells disappear, or their concentration falls and becomes too low to maintain the normal effective immune response. 2 words about fungi. Fungi may occur as the ectocellular or intercellular parasites. For example, Histoplasma capsulatum, what's shown here, with the photographs in the center, may occur as the intercellular parasite. In this case, Histoplasma capsulatum has the living form of the yeasts, and these yeasts may parasitize cells and accumulate inside of these cells. Of course, immune response to fungi is the combination of the humoral and cellular immune responses. Now about innate immunity. Neutrophils and macrophages are the major cellular components of the cellular innate immunity. Neutrophils and macrophages at the surface have many receptors for fungal pumps, pathogen-associated molecular patterns. They are Toll-like receptors, Dictin-1, Dictin-2 receptors, Minkel receptors, the macrophage receptors, and receptors for mannose residues. All these receptors, when bind to the fungal substances, start functioning of the activating pathways that activate transcription from the certain genes. Transcription, for example, of the cytokines that maintain development of T helper type 1 and T helper type 17. It means macrophage is activated by pumps via what binding to receptors that are shown here. Started producing of the interleukin-12, for example. Interleukin-12 stimulate maturation of naive T helper cells to the T helper cells type 1. And second, help to differentiate T helper cells from naive to the T helper 17 types. For this helper 1 and 17, they are very important in the antifungal immune response. Very interesting and important mechanism of the immunity against fungi is the formation of so-called neutrophil extracellular traps, or NETs, N-E-T, net. Process of formation of neutrophil extracellular traps is named netosis. Now, why this process is activated? Usually fungi are large. They are larger than bacteria. And they may be too large to be engulfed by the phagocytotic cell. In this case, one fungal organism may be attacked by the group several neutrophils. And these neutrophils try to digest fungal molecule, fungal body, but in some cases the digestion becomes low active. Now, what are the substances that neutrophils, for example, macrophages, try to produce to digest fungal organisms? The oxygen-dependent substances, nitric oxide, for example, hydrogen peroxide, hypochlorite radicals. In some cases, only these capacities are not enough to destroy fungal organisms. For neutrophils, here being described the very interesting process that facilitates digestion of fungi and prevents their spreading in the host organism. This is netosis. What is happening? Neutrophil activated by the attempt of the digest fungal organism releases own genome, own DNA from the cell towards the fungal surface of the fungal cell or fungal hypha. This DNA molecule is very sticky, binds to the surface of the fungal organism, and usually sticks to the surface digestive enzymes that may destroy fungal cell, for example, elastase. What is happening with this neutrophil? Of course, neutrophil dies, because neutrophil with the DNA genome cannot live. But DNA released from neutrophil during formation of nets, realization of netosis, this DNA is not ruptured. You know that in some cases cells undergo to the apoptosis and die because DNA is fragmented and destroyed. In the netosis, DNA is not fragmented and destroyed. This is a very long last molecule that is undestroyed, and in this form coats all the surface of fungus, forming barrier to this fungi to be released. And from other side, when this DNA binds elastase and some other enzymes to the surface of fungi, and these enzymes digest fungi. What in this slide you see in the lower part, in the photograph E, how DNA that is shown blue, DNA of the neutrophil binds to the hypha of the pseudohypha of the candida. This is a fungus. Candida is shown red. Nucleic acid sticks to the surface of this red fungus and combines, not combines, and sticks, facilitates binding to the surface of fungal of the neutrophil elastase, shown green. Elastase, this is a very powerful digestive enzyme, destroying different types of fibers, including cytoplasmic fibers of fungi, and fungi organism dies. It means netosis is a very important mechanism of elimination of the fungal organism, and we know now that netosis may be directed against bacteria also. Bacteria and some parasites. No, innate immunity to fungi, of course, consists of many components. Here you see complement activation by Aspergillus organism, but many fungi may neutralize complement activation because they may produce complement inhibitors activation of complement system. Fungi may secrete proteases that degrade such complement proteins as the C3b, C5b, or C9, and this way neutralizing complement activation system. About adaptive immunity to fungal cells. In the adaptive or acquired immunity, also cooperation of the antigen-presenting cells, T cells, and the T helper cells and T killer cells happen. So, engagement of dictin-1 and dictin system of receptors is a very important system of receptors of the many human cells, including dendritic cells and leukocytes. If extracellular fungi they bind to the dictin-1 receptor, the dendritic cell after that start producing pro-inflammatory cytokines, interleukin 1, 6, and 23. And these cytokines, in the presence of interleukin 12, they cause the maturation of naive T helper cells to the T helper type 1, in some cases to regulatory cell. And the cytokines interleukin 1, 6, and 23 with these pro-inflammatory cytokines, they make T helper cell of producing interleukin 21. And what is happening? In the lymphoid organ, place of inflammation, accumulate T cells producing interleukin 21, and this is a cytokine of the T helper 17. And this T helper 17 start producing interleukin 21, supporting maturation of naive helpers to the Th17 cells, 17 cells. Additional stimulation of the transformation of naive T helper cells to the T helper 17, matured, component cells, is the secretion by different cells of the transforming growth factor beta that in the complex to interleukin 21 stimulates differentiation of T CD4 cells to the T helper cells of the 17th part. Th17th cell is the important part of the cellular antifungal immune response. So, a real picture is more complex. Dictin receptors may react to the extracellular fungi, stimulating dendritic cells for the production of pro-inflammatory cytokines responsible for polarization of CD4 precursor cells to the T helpers type 17. Also, with this differentiation of the naive CD4 cells to the T helper 17 is stimulated by engulfment by the antigen-presenting cells of the apoptotic fungus-infected cells. Apoptotic fungus-infected cells also stimulate dendritic cell and macrophages in the production of the interleukin 1, interleukin 6, and interleukin 23. In the same time, some of the descendants of T helper naive cells in the presence of these pro-inflammatory cytokines, transforming growth factor beta, and presence of interleukin 21, or these cells, naive cells, more and more actively start to be transformed to the T helper 17. T helper 17 produce a cocktail of cytokines, interleukin 17 alpha, interleukin 17 F, interleukin 21, interleukin 22, tumor necrosis factor alpha, interleukin 25, interleukin 26. But all these interleukins stimulate so-called neutrophil inflammation because the cytokines facilitate recruitment of the neutrophils from blood and lymph stream towards the place of infection, activate neutrophils, and they start to attack and digest fungal cells more effectively. It means fungal acquired immunity is associated to T helper type 17. And this maturation is stimulated by engagement of dectin-1 receptor in the recognizer fungal pumps and phagocytosis of the apoptotic fungal cells by the dendritic cells. In this scheme, this is the last slide in my today's lecture, you see complete picture demonstrating activity of different cells in the struggling against fungi. First of all, in the left part, at the top you see epithelial cells with epithelial cells with the cilia on the surface. In the upper left part you see how candida with fungi contact to the macrophage with a dendritic cell, well, this is a dendritic-presenting cell. They become digested and stimulate produced by this antigen-presenting cell of the pro-inflammatory cytokines, interleukin-1 beta, tumor necrosis factor, alpha and beta, and interleukin-6. We can add to this list. Interleukin-12, interleukin-23. These cytokines activate influx to this place of the neutrophils, what you see here, 3 neutrophils. They produce ROS. This is a reactive oxygen species, including hydrogen peroxide and hypochlorite. And what neutrophil passage from the vessel to this area stimulated by this interleukin-1 and interleukin-6. This neutrophil try to digest fungal organisms, and one of the methods is mitosis. You see what third low neutrophil shoots the fungal cell by the digested complete DNA. DNA binds to the fungal, stick to this fungal what green balls are the elastase that stimulate digestion of the fungal organism. In the same time, dendritic cells in the lymph nodes and other immune organs stimulate, because they occur in the presence of the interleukin-1, tumor necrosis factor, interleukin-6 produced by other dendritic cells or macrophages. Well, these dendritic cells engulf fungi and produce cytokines facilitating development from naive T helper cells, zero cells, the helpers of the 17th type and helpers of the type number 1. Dendritic cells secrete interleukin-12. They secrete interleukin-6 and interleukin-1, and this way maintain the transformation of naive cells to the T helper 17. T helper type 17 produce interleukin-22. This interleukin-22 binds to the ligand to the interleukin-22 on the epithelial cells, and they start producing antifungal beta defensins, substances that destroy fungi in the lumen of the organ. Additionally, T helper type 1 cells also are matured because interleukin-12 produced by the dendritic cell and macrophage also helps naive cells to be transformed to the T helpers type 1, as they produce interferon gamma, activating macrophages and activating dendritic cells for their functions. Time of the lecture is over. Thank you for your attendance, for your attention. I hope that my today's lecture will help you to distribute your knowledge about bacteria, viruses, and fungi better than you distributed these things before. Be healthy. Have a nice weekend. Goodbye. Мгм. 2 человека. Ну что ж, это тоже кое-что. The lecture is finished. Thank you.